Hi, I want to say good afternoon, but I realize for most of you it's actually good morning, or maybe good mid-morning. So let me jump straight into it. So this is a ZP live streaming webinar um, discussing screen-printed electrodes, but we've got lots of demonstrations as well. So I just want to kind of share a screen with you, or a camera with you rather, just to kind of give reassurance that this is not your typical webinar with just slides and people talking in theory. As you can see, this will probably jump up at you now, but I've got potentiostat in front of me. I've got at least two types of screw-printed electrodes here. The can of Red Bull is not because of the requirement for energy. It's just to kind of illustrate some, I want to say things. But that said, let me go ahead now and do the webinar. So that's just to give you a sense of what you can expect. They'll be mixed up. I'll go fairly fast. Time is most valuable asset we all have. So I am going to touch on the fact that, why should you even care about screen-printed electrodes? So that will be definitely something I cover. I'll talk about the glucose strip, but some of this is stuff that you will already know, but I will just go through it fast. I will talk about screen-printed electrodes. I will talk about practical real-world examples of screen-printed electrodes, and I'll also do some. In total, I'm going to end up doing, I don't know, at least 6 experiments, somewhere between 6 and 10 experiments, but they'll all be done fairly fast, which is really the power of, let's say, electrochemical analysis and screen printed electrodes. So I will use some Sudan Red. I will touch on ferricyanide. I'll touch on Randall-Sevich equation. As I say, even this can of Red Bull is going to feature in our real world demonstrations of screen printed electrodes this afternoon. So why care? The why care part, I know that some of you, well, many of you are actually already users of ZP electrodes. Why should you care about screen-printed electrodes? And it's the classic kind of glucose sensor. When they started their life, they were actually, they were all printed. And I think the glucose strip really demonstrates the utility of screen-printed electrodes. So let me just stop sharing that screen for a second, and let me just change here and just do this very quickly. Right. So let's say I was pitching to you the idea of that I wanted you to adopt screen-printed electrodes. So I would say, okay, screen-printed electrodes have really proven themselves to be an ideal tool to make low cost, easy to use, and rapid biosensors. And if I was to demonstrate it, I would take a glucose strip like this, which were historically produced by screen printing. I'd put it into a device like this. That now demonstrates, what that's demonstrating is both that, electrochemistry or screen printed electrodes are a good choice for scaling electroanalysis. And actually, electrochemistry in general is a good technology because you can make these low cost meters. Now, what I'll do is demonstrate the sample prep, which electrochemistry, so the sample prep is take some blood. So there's no sample prep effectively. And it also illustrates something else about electrochemistry as well, is in fact, I don't need much sample. I mean, that's 300 nanoliters. And I've got 99 milligrams per DL. A lot of you are in North America, so that would be the kind of units that they would use there, 99 mg per DL. In Europe, they may call that more like 5.1 millimoles of glucose. But that illustrates everything that I would want to say about why you should even care about screen-printed electrodes. That product and that technology stack, I think, more or less illustrates the point 100%. There's billions of such technologies done per year. And if you're looking for something that will scale your science out of the lab, then screen-printed electrodes and electrochemistry is is a good tool for you to do that. If you're attending this afternoon, by the way, I will. This is being recorded, and at the end, I'll send you a recording later on. Now, what we're doing at ZP is actually taking that good workflow that you have in a glucose strip and then putting it onto a new type of potentiostat. Now, the main focus this afternoon is actually on screen-printed electrodes, but I will be using this Sensi or potentiostat from ZP as well to illustrate some points. So what does the glucose strip teach us? Well, it teaches us that it can be low cost. this kind of technology can be low cost. The reason I say can be, because the reason they get such low cost when they're making glucose strips is because they're basically producing billions per year. And when you produce, the higher your volume, the lower your cost per item, and actually the better quality per item it is as well. So high volume manufacturing does drive down costs. What does that short demonstration where I'm just measured my blood glucose show you? It shows you it's easy to use. And I do want to illustrate that if you're developing assays using screen-printed electrodes, they should be easy to use. Sometimes when you read scientific papers, people are often normalizing data, or people are having some sort of strange calibration. Now, they're doing that because there's a problem with reproducibility in screen-printed electrodes. But I and now some of you are users of our electrodes, so you can probably, when I say call me out on this, I do try to support you then and sort of explain to you where your irreproducibility might be coming from. But I will demonstrate to you a real world example, and I will get a really good result because of the reproducibility of the electrodes. And then what else did I just demonstrate with that quick bit of bloodletting where I just read my blood glucose? It was a rapid result, both in sample prep and test time. Electrochemistry, I feel, is one of the few science, it really wins down when it comes to sample prep. That I didn't prep my blood in any way. I just put my blood on that strip, and that was enough to make an electrochemical measurement. Most analytical techniques would require some sort of filtration, purification, enrichment maybe of that sample before reading, but that's not the case with that blood glucose meter. Let's dive a little bit deeper now onto the screen-printed electrodes. That was the title of this. So at ZP, we have a kind of lots of electrodes, lots of electrodes, but I want to kind of highlight a few things, just FYI. In the US, we do have a distributor called IO Rodeo, and I know some of you buy from IO Rodeo. I mention it because the discount code that I'll give at the end won't work for them, but it's not a bad place to buy our materials from, IO Rodeo in the US, because they basically buy in bulk and they put it into smaller pack sizes, which can be quite useful for you. And also they bought the materials. They bought the materials a while ago, so it means things like tariffs weren't in place at that time. Now, materials that if you are starting a new program soon, you've got a choice of materials when it comes to screen-printed electrodes. Carbon and graphite. Sometimes I call them carbon, but strictly speaking, many of our screen-printed electrodes are really graphite. So, these two guys that I'm holding up, carbon and graphite. Those are my personal favorite materials to use when developing assays. Now, you've also got the choice of gold, platinum, and graphene. And there's probably more as well, but these are the kind of four main materials these days: graphite, gold, platinum, and graphene. So let me first of all just say about gold. At this moment, gold prices have really spiked up. So please do not develop assays on gold. Why? Because gold is a precious metal, and when you're trying to go for a low cost sensor, you can't be putting gold on it and think that you're really going to help yourself with that low cost claim. Even though you may not be using much material, the gold price, I think, has tripled in the last few years, and in the last few months it's spiked again. Gold is, when the economic situation globally is uncertain, people rush to gold. People buy gold. You're competing with people who are investing in gold. Gold is never going to be low cost, and because it's never going to be low cost, it's never going to lead to what you're really looking for, which is, I would like to develop a low cost assay. Well, don't use gold. You can always do it with carbon. Now, the reason people use gold is because of self-assembled monolayers. They're very used to many tens of thousands of papers, if not hundreds of thousands of paper, that talk about self-assembled monolayers, where they take the gold electrode, they put the sulfur atom on there, and it forms a strong bond. That's great. Self-assembled monolayers are great, and that's migrated over to biosensors, and so everyone wants gold electrodes, but gold is expensive. It's tripled in price. It's not coming down anytime soon, and it does not marry well with what everyone's trying to do, which is make low-cost biosensors, low-cost assays. So please don't use gold. I'm a little bit softer on platinum because I understand that platinum actually has some catalytic properties. So, for example, if you're making an enzyme sensor, glucose, lactate, alcohol, many of them are oxidases, and oxidases produce hydrogen peroxide. And so it's fairly simple to take an oxidase enzyme that's specific to your analyte. The oxidase reacts with your analyte, it reacts with the cofactor, which is oxygen. Oxygen becomes hydrogen peroxide. So I understand platinum. It's that catalytic surface that you're after. So I'm a little bit softer on platinum, though I do find that actually the materials that people most request from us are carbon and gold. So 60% of you are either are interested in carbon or gold. If I can slightly bias you, always go for carbon. It's a fraction of the cost. It doesn't compete with other people who want to buy gold because they want to protect wealth, and that's why people buy gold. Platinum I'm a little bit softer on, but I think with people using screen-printed electrodes, platinum's less of an interest anyway. And then graphene is, it's not the material that people otherwise would like it to be. Now, why is graphene popular at the moment? Well, the reason graphene's popular at the moment is because when a material is novel and you want to be published in scientific journals, you have to just demonstrate that you're novel. And so, yeah, I'm using graphene, that's novel, but I think that ship has sailed at this point now. Everyone knows graphene. The Nobel Prizes were given out a few years ago. Graphene's a material looking for application, but I don't think it adds a lot of value to biosensing. There's claims, but those claims, in fact, I can't think of any real commercially successful graphene-based sensors. There's plenty of people doing that work, I get it, but it doesn't really add that much technical advantage. And I'll show you some perfectly good cyclic voltammetry, not using graphene this afternoon. Be very careful about bespokeness. I was doing a webinar the other day. So here's a sheet of electrodes. Here on this sheet, before I cut out a few of them in the corner, there were 300 electrodes. Now, the reason I hand that up is because people will often say to us, oh, well, I need a unique design. And, I'll say, okay, how many do you need? And they say, I need 1,000. 1,000 would be 3 sheets of this. Well, that would be 900, but give or take 100. So you can understand that when you're asking for something bespoke and special to you, unless you've got large volumes, I could give you 1,000 or 900 in just 3 sheets. And you may say, oh, I want 10,000. Okay, well, that's 30 sheets. These are tiny manufacturing runs. It's hardly worth them turning the machine on to actually produce 30 sheets. Now that means it's not really manufacturing. It's not manufacturing. So don't be very careful when asking for custom designs and bespokeness, because the volumes you're asking for are so small that it's not really manufacturing. And what it leads to is you want low cost and people will give you low cost, but what they won't tell you is actually, because we're hardly manufacturing this, it's actually going to be low quality. So you get low cost but actually get low quality. That low quality then spills into your research and you find, oh, I've got irreproducibility between my experiments. What's going on here? It's the underlying electrodes. Now, we do have good reproducibility because we're actually producing at volume. So be careful with bespokeness. Most of our designs actually take care of compliance voltage. So I'm sure you're more than aware that potentiostats are getting smaller. The little video that's playing in the background here, this is one of our latest wearable potentiostats. The reason I sort of show that is because when you've got potentiostats like that, they're very low power. And so ZP, I think, is one of the few companies that really understands something called compliance voltage. And so when we design screen-printed electrodes, we're designing them understanding that even if we're using potentiostats like this these days, and I'm just going to hold up the sensor or potentiostat, this is USB-C driven. So everything's becoming lower power. So our screen-printed electrodes are one of the few ones where we've actually taken account of the fact that potentiostats are becoming lower power, and it takes care of something called compliance voltage. I have done videos on compliance voltage, so go onto the YouTube channels or ZP and compliance voltage, and you will find a discussion about compliance voltage. And then the other thing that really messes people up with screen-printed electrodes is connectors. I'm going to make some perfectly good connection to the screen-printed electrodes this afternoon, but a lot of these connectors where you're kind of slotting this thing in, it leads to high impedance connection. And I think it really, when you've got high impedance to your screen-printed electrode, it makes the screen-printed electrode look bad. You think, oh, it's the screen-printed electrode, it's the carbon ink or something like that. It's actually not. It's the electrical connection between the potentiostat and the screen-printed electrode that's the problem. And I do have, again, videos on that. I also do a weekly webinar, so if you've got technical questions, send them in and we kind of cover them in those weekly webinars. But let me go forward a bit more now and give you a practical example. So I would like to persuade you that screen-printed electrodes and electrochemistry are absolutely fantastic at doing commercial assays. So let me not talk about it. Let me just show you an example. So I have a sensor or potentiostat in front of me. I have the sensor or app in front of me. It obviously says caffeine on that. Let me pull in a can here and let's ask the question, how much caffeine is in this Red Bull? So we can ask the question. You can actually read it on the can, but let's take the electrochemistry approach. And one of the things I love about electrochemistry is this. I'll take this sample and I'll put it in this vial. This sample is from a carbonated drink. So this means it's actually got bubbles in it. That would really mess up an optical assay to have those bubbles in that, but it's not going to mess up the electrochemical assay. So let me just put that down for a second, move that out of the way. Let me just grab, I've grabbed 1 volume of the carbonated drink, which has got caffeine in it, it's Red Bull. Let me grab 1 volume of buffer, because most of you have a strong background in electrochemistry. You can understand that carbonated drinks don't have a lot of electrolytes in them. So we do have to add those electrolytes. Now let me show you the next power of electrochemistry. All right, so I've put those 2 volumes together. Let me do the sample prep. Done. Right, now let's take 50 microliters. That was the other point from the glucose test I did earlier on. Don't need much sample. Now in the world of food testing, 50 microliters is not, people don't, people don't worry so much about 50 microliters in the world of food. But in the world of blood testing, and patient samples, actually they do care. So I'll put the sample on like that. I will hit, let me just name it in the app so we can find it later on. Red Bull. Done. Right. Continue. Start measurement. Now this is running square wave voltammetry. So that's just the baseline. In a minute, you'll see the signals change significantly. So here's the signals starting to change significantly. And here comes the caffeine peak now. And there's the caffeine peak. You did notice a few things earlier on. Those are actually extra molecules, theophylline or theobromine or something that is also in caffeine as well. So what it's now doing is it's uploading the data to a cloud system called Julie. I know some of you are already familiar with our kind of technology stack, but I'm also going to share my screen with you so that you can see the data go to the cloud as well. So I've got a result on my phone. Just before I come off here, if you look on the side of a can of Red Bull, you will see that it's 0.03% caffeine. 0.03% caffeine. 0.03% caffeine translates at 0.3 milligrams per gram of caffeine in that drink. So I've got here 0.261, but take 0.261 to 1 decimal point, that's 0.3 milligrams per gram. So that really talks about the reproducibility. I can do this assay all day and give you the same result, which is talking about the underlying reproducibility of our screen-printed electrodes. Now I just had a message come through and I think it was about stability, so I'm going to take a quick look at it. Yeah, okay, I can answer these questions, no sweat. Okay, so I just want to make sure I've acknowledged those questions. Now what I want to do is I just want to share you this and talk about that data went straight to the cloud. So this is Julie. This is probably one of the, I think it's, if somebody knows better, then let me know. So I think this is one of the few or only cloud-enabled potentiostats actually on the market. So there's the Red Bull. Let's have a quick look at that. So never ask yourself the question, where is my data? This is really a data science tool as well, because what we've got going on here is, so this is the time and date, universal time. So in fact, it's 4:18 in London, but it says 3:18 because it's universal time. The date, me, because I did the work. This is the square wave voltammetry. There's some interferences, but there's the main signal. Look at the way we can process the signal to take care of baseline. So we've baseline corrected. Look at the way we've done feature extraction using this system. Although this comes with a potentiostat, by the way. And then we've found the feature, which was peak height in this case, and turned it into milligrams per gram of caffeine. Now, the reason I did all that was because a minute ago when I was doing practical example, I was saying, okay, well, let's not talk about why you should be interested in this technology. Let's do a real world example. That was done live for you, so it talks somewhat about the robustness. So I took the sensor or potentiostat. I put one of our sensors onto it. I put the sample onto it. The app, which is available on the Google Play Store and the Apple App Store, I just scanned a QR code. That put it into the right modality. The Bluetooth connection between the smart device and the potentiostat. So I sent via Bluetooth the commands. The instrument ran, the data came back to the instrument. The instrument actually sent the data in real time to the cloud, which I just accessed, where we processed it, we analyzed it, and we sent the result back to the phone. So when I got that result on my phone of 0.261, that whole turnaround was actually from my phone up to the cloud, processing the cloud, and back to my phone as well. So we definitely moved on from the world of glucose strips, where that kind of, when these things were first invented, that whole workflow was not even possible. But we've reimagined what a glucose strip could do, but imagined it for 2025. Now I'm going to do a few more demos here as well, and those demos will help. I'm just going to read the question. Could I explain stability of SPEs in terms of range of temperature and acidic conditions in which they maintain their sensitivity? I'm actually going to use acid this afternoon on these screen-printed electrodes. Temperature range is interesting because the screen-printed electrodes that I am using this afternoon are actually manufactured at 150 degrees Celsius. I do not want to promise you an operating temperature of 150 degrees Celsius, but I do want you to understand if they're manufactured at 150 degrees Celsius, obviously most aqueous systems will boil at 100 degrees. So I suspect that the operating temperature is actually well above that. We also make them out of ceramic as well, where they're actually processed at 700 degrees Celsius. But I'm also going to answer your pH question as well. So I'm going to do 3 tests this afternoon: Sudan Red, Ferricyanide, and I'm also going to do some Randall-Sevich analysis. So with Sudan Red, I do have some Sudan Red here. It's an azo, it's a diazo dye. Is it an azo dye? No, it's an azo dye, not a diazo dye. It's an azo dye. I will do the oxidation of this azo dye. This is what we call an EC mechanism. The reason being is that the azo group here is electrochemically oxidized, and it's cleaved. And the two products then can't just come back together. So it's effectively irreversible. So we have this electrochemical event, and then we have this chemical bond breaking. So we call that an EC mechanism. Now, what I want to do is stop sharing for a second, which I will do. I'll change cameras and I'll do all this fairly quickly in front of you. So I will grab a sample of an azo dye, which I am doing. I will dispose of this electrode. I don't want to say this, but I'm actually starting to realize as well that sometimes you can reuse electrodes. You'll see me reuse electrodes quite a lot this afternoon, especially in a teaching kind of environment. In a research environment, you kind of have a lot of things going on an electrode. But in a teaching environment where actually the electrodes may be only doing one piece of chemistry, like the oxidation reduction of ferricyanide, you may be able to actually recycle or reuse, I should say, not so much recycle. All right, what I'll do now is I'm going to go into a slightly different mode. So I'm just going to grab some QR codes. I will scan cyclic voltammetry. So now I'm in my cyclic voltammetry mode. I'm going to call this Sudan Red. Sudan Red demo, yeah. I'll set the start voltage at minus 200 millivolts. I'll go to 1000 volts. I'm going to go at 100 millivolts per second. I may set that to 100 microamps. All of this is done live, by the way, so forgive any kind of errors that I make as I go along. And I will now take the sample. Again, 50 microliters, fantastic. 50 microliters onto the sensor. I hit start measurement, and it's effectively now running that scan. I use 100 millivolts per second because I want my assays to be fast. Doctors and nurses do not want to wait around for an assay to run. So if you've got the choice of making, a slow assay or a fast assay, I would always choose to make my assays intrinsically fast. So now it says the upload is complete, and I'm just going to quickly, I'll go done there. I'll share my screen with you. We'll flick over to, that was the caffeine data. Let's go up there and find that Sudan Red data. So let's just do a quick refresh here. There it is. And when it pops up, which it will, there we go. I can see the oxidation of Sudan Red, but very little evidence of a reduction of Sudan Red. Why is that? Because Sudan Red has an EC mechanism. So I just want to kind of demonstrate a sort of non-reversible system on a screen-printed electrode. Now what I want to do is quickly go to here and, whoops-a-daisy, I'm going to do a classic experiment, ferricyanide. Ferricyanide is actually quite useful, and we use it a lot in our assays. It's quite predictable, it's quite stable, quite easy, quite non-toxic, quite easy to get hold of. Classic cyclic voltammetry of ferricyanide, but I'm going to do it slightly differently. So I'm going to gather the oxidation reduction of ferro to ferricyanide as it sort of almost like a classic cyclic voltammetry. I'll have a beginning, an initial potential. I'll go to the final potential, and then I'll go back to the initial potential. Now I'm going to do all of this. Whoops a day. I realize I'm talking to you, and I'm not actually sharing my screen at that point. And maybe I didn't share my screen. I haven't been sharing my screen for a little while, so I might just slightly go back and just kind of apologize. That was the Sudan Red data, by the way. So Sudan Red, I got the oxidation. I didn't get the reduction. I didn't get the reduction because it's an EC. It was an EC mechanism, so the C part followed the E part and stopped me having reversibility. Thanks for the heads up, but I appreciate it. You can see this is live. Now I'm going to do the cyclic voltammetry of ferricyanide. So ferricyanide, I've got that classic voltammetry. We have a start potential. We're going to oxidize ferro to ferri, and then we'll take ferri back to ferro by using a triangular waveform like this. But thanks for the heads up again. It is appreciated. And I will, I'll probably use these conditions, which I'll run through very quickly. I like fast assays, so 19 seconds, start potential -200, turn potential, turn voltage 600, fast scan rate, only one repeat, 100 microamp range. I'll take one of our screen-printed electrodes, and I will put a drop of ferricyanide on there. I'm going to do something a little bit different, though. What I'm going to do is this. I'm going to record one without cleaning, then I'm going to clean it, and then I'm going to do the cyclic voltammetry again. So what's not talked about, but thanks to the interaction with some of you, I have now started to promote the idea of cleaning screen-printed electrodes with acid solutions, and I think they withstand acid solutions really quite well. And how will I make you believe that or not believe it? I will just show you. So I'm about to do 3 experiments, or 2 experiments and a cleaning step. So without cleaning, clean, and then I'll clean, and then after cleaning, we will have a look at that data. So thanks for the heads up about not sharing the screen, but it does prove that we are live. And I will also then go to the desktop here. So what I'm going to do is, I'm going to take that off in case I didn't put a clean electrode on, but I'll put a clean screen-printed electrode on. That's on, great. Now I'm going to take some ferricyanide. So remember now, this is not a criticism of Zimmer and Peacock, but I actually think, I realize that when we're manufacturing, when we're manufacturing a new assay on top of a screen printed electrode, I realized that we actually often have in our manufacturing process, take electrodes, clean electrodes, functionalize electrodes. I just realized now we just need to kind of promote that's actually what we do. Everyone loves, not everyone, but lots of people like glassy carbon electrodes. The first thing you do when you pick up a glassy carbon electrode is you actually clean it. So there we are comparing screen printed electrodes to glassy carbon electrodes, but we're not being fair because actually you cleaned that glassy carbon electrode just before you used it. Let me just change this to before Ferry so we can find it. I will try and go fast because I appreciate, you know, people can't hang around forever. So before Ferry, that just means I'm before cleaning basically. I'll go continue. So I'll start at minus 200. I'll go to 600. I've got 100 millivolts. I think everything else is good. If I make a mistake, my apologies. I'll just go start measurement now. So we'll get the oxidation wave. Now, the reason I get the oxidation wave for oxidation of ferro to Ferri is because I started my solution, which is Ferri, at minus 200 millivolts. So I actually I started by the reduction of Ferri to ferro, and then doing the voltammetry, the Ferri was then the ferro was then reoxidized back to Ferri. Now it says here, upload complete, okay? Right, now I'm gonna clean this electrode with an acidic solution. Now I'm going to use pH 2. I'm just using what I basically have at hand, but I'm going to use pH 2. I'm pretty sure you can get away with pH 1 as well, but I will use pH 2 because that's what I have at hand. So let me take, let's clean this electrode now. Let's activate this electrode. Not a terrible thing to do because in fact we do activate our electrodes when we're manufacturing products upon them. Now what I'm going to do is go done. I want to change the conditions a little bit. So this is the cleaning. Cleaning, right, cleaning is said, done, good. I want to go continue. I'll start at minus 1 volt. I'll go to plus 1 volt. I'm only going to do one cycle. This will just take 43 seconds. Don't develop procedures and stuff that take a long time to do. You can't, it's an MIT philosophy, that one's got to fail fast. So you don't want to be waiting around a long time to do scientific experiments. I know sometimes science does take long, but in this little domain of electrochemistry and the use of screen-printed electrodes, I can understand if you're doing a 3-day experiment, that's one thing. But if you're doing cyclic voltammetry and developing point of care assays, if it takes a long time to run, then it's both a drain on your time, but also imagine that one day somebody's going to try and use that assay in the real world, and you say, oh, how long does that take? 20 minutes. 20 minutes, the patient's left by then. All right, so I've done the cleaning step now. Now you'll see me take that acid off that electrode, and I will then bring 50 microliters of the ferricyanide back to that electrode, and we'll do a final, let's say, experiment on that. So here it goes. So 50 microliters onto that, which is done. I'm just going to change the conditions back again. I'll call this after though. So I'm just going to call this after, and then I'll call it ferricyanide, just to remind us that that's actually, what that is. I want to start at minus 200 millivolts, which I've done. I'll take that to 600 millivolts, which I've done. I'll keep the scan rate at 100. I think everything else is good. I'll go start measurement, and off it goes. So oxidation of ferro to ferri, there goes the oxidation wave now. Then there'll be a reduction of ferri back to ferro. Here it comes, and then it will finish the assay. I say upload complete, and the data's gone to the cloud. Right, I will remember this time to actually share my screen with you. But again, thanks for the heads up. It's appreciated. What I will now do is say, okay, that was that Sudan Red, but since then we've done 3 experiments. And I'll do a refresh because everything will be up in the cloud. Right, so we got before, cleaning, and then after. So let's have a quick look at before. So I can see some cyclic voltammetry there, nothing super spectacular. I'm just going to look at after, and then I'll do a comparison as well. So you'll be able to sort of see side-by-side comparison as well. Right, okay, let's have a quick look. Now, maybe there's not that much difference between these two actually, but let's have a quick look. All right, so before and after, and I will combine these two just so I can have a quick look to see if there's been an improvement. So that's brought the two together. Now I'm just going to go to tools and properties and discrete values, and let's just have a quick look at what we've got. Yeah, so to be fair, there's not actually that much improvement on that occasion, which will teach me for doing a live demo, but I think it's because both CVs are not that, in fact, not that bad. So before and after, I've tried to improve it. There's a little bit of improvement on the reduction, to be fair. That peak has gone from here to here. I've got a slightly bit more current, so there's a bit of an improvement, but there's not that much improvement. Well, yeah, I mean, it's a bit sharper up here, but I would say it's quite marginal. What it does talk about is the reproducibility, that the electrode has not drifted. So I could argue that the acid has marginally, in this particular case, improved that electrode. But it didn't certainly damage it and make it unusable, if I can put it that way. So now what I want to do is, I'm coming closer to the end. I can see somebody's typing a question, so I will try and pick that up. I just want to talk about Randall-Sevich very quickly. This is a bit of a classic. I do have some videos about Randall-Sevich. In the Randall-Sevich, it kind of says when you're doing something like ferricyanide, the peak height is proportional to the square root of scan rate. This comes from fixed laws of diffusion, by the way. It's not just magic. It's just because the peak height is often under diffusion control, and that's where this kind of current versus square root of scan rate comes from. So what it says is that the peak height is proportional to square root of scan rate, and if we plotted peak height versus square root of scan rate, then we should have a linear graph. What I want to do is I want to do a demonstration where actually I'm going to do a series of experiments pretty quickly. Don't worry, you're not going to be hanging around, but I just want to show you the kind of efficiency of this technology stack and also screen-printed electrodes as well. So I'll stop sharing that for a second. That will make my screen jump up a little bit. I'm going to show you the last QR code of the day, which I will scan. So I've scanned that QR code. I've put myself into a particular modality. I'm going to reuse that screen-printed electrode. I didn't need to necessarily throw it out this time. I'm going to give this a name so we can find the data. Randall Sevich. Terrible spelling, but not terrible spelling, terrible typing. Right, and then I'll go continue, start. Right, so this will be fairly fast for you. What I'm basically done is, with that QR code, this is the power of this particular potentiostat, that with this, with that one QR code, I've actually set up 4 experiments to run sequentially at 4 different scan rates, probably something like 50, 100, maybe 200, and maybe 250. It's running them now. That's the first one coming to an end, then it'll automatically progress to the next one. So there it goes, the next one. You'll see that these assays are becoming faster and faster because the scan rate is going faster and faster. So we're expecting the peak height to be proportional to scan rate in line with the Randall-Sevich equation. So that is a bit bigger now. And then the third and the fourth one, I think, will just absolutely fly through. Again, I love that. Look, a 10-second sensor. People don't appreciate that. I understand that voltammetry gets distorted by scan rate, but it definitely improves the user experience to have faster scan rates. So this one is only about 7 seconds long, and then it's done that. Don't forget, now it's gathered 4 bits of data, sent them to the cloud. Can you imagine how much time people tend to take to process this kind of data? I'm just going to share my screen with you again, which I will share here. I will change that here. Right, so let's have a look at this data. So reports, cancel. I'm just going to say that for a second. I just want to save what we did in that particular report. And then I will go here and I will do a refresh here. And then the Randall-Sevich data has just jumped up, which is doing now. And then you'll find out that we've got 4 sets of data, 1, 2, 3, 4 different scan rates. And it's also processed the data and fitted it to the Randall-Sevich equation. So I've got essentially here my current is proportional to we've done it to the log there. Here it is. Yeah, I was going to say we did it to the log there, but we did it to the square root of scan rate. So that's also showing you that now people are they like to do this experiment. The reason they like to do it is because it's actually quite straightforward. And I think I've just demonstrated how quickly you can do it. So we think that's quite a nice experiment for actually doing it in the teaching lab because it was quite easy to do. So what I want to do now is probably finish off and just say, so today we've talked about screen-printed electrodes. I did have some live questions coming through. I can see a long question being written, but it keeps disappearing on me. Screen-printed electrodes. Somebody was just asking here if I recommend cleaning the sensors before use. When Zimmer & Peacock is making biosensors these days, when we're trying to make biosensors, we've got this manufacturing process where we take screen-printed electrodes, we clean them, we then functionalize them. So I would actually say that it is a good idea to actually have a cleaning step in there. So today, and if you want beautiful data, like, people like to use glassy carbon electrodes because they give beautiful data. But of course, what we're all forgetting is, it's not because glassy carbon was actually so fantastic. It's actually because it's been cleaned just before use. So we've discussed screen-printed electrodes today. We've definitely discussed, that this is something that if you want to make commercial products, the glucose strips definitely prove that out. I did you a live demonstration of Red Bull. You can check up the milligrams per gram, or the milligrams per gram of Red Bull is 0.3 milligrams per gram. That's what we got, or it's 0.03% on the side of the can. You can check that out. We did a Sudan Red, which was an EC mechanism, a ferricyanide, which is just an E mechanism, reversible. And I also gathered Randall-Sevich data probably in less than 2 minutes and got it all processed in less than 2 minutes as well. So there is a discount code. Now, most of you are actually, a lot of you are in the USA. Now, the reason I bring that up is because actually IO Rodeo is not a bad place to get our electrodes because you can get smaller pack sizes. They've already taken care of the shipping, etc. So unfortunately, this discount code won't work on there, but you can use the discount code on our web store, which is shop.zimmerpeacock.com 10%_May.2025. That will be the discount code, but you'll get a recording this video anyway, so don't essentially sweat it too much. I can see a question coming through, but I am going to get close to saying thank you now. Oh, right, that's really cool. Yeah. Right, somebody's asked a very good question at the end here, and I will answer it, which is, can I comment on the stability of surfaces with non-aqueous solvents like acetonitrile, DMSO, etc.? And the quick answer is those, are very, they're pretty aggressive organic solvents. They absolutely will essentially dissolve the material. So these kind of electrodes, we've got experience that I think the most organic thing that we kind of get away with is actually 5% ethanol in aqueous. I know that's low, but that's what, strictly speaking, lots of screen-printed electrodes on the market, including Zimmer Peacock, are not super resistant to organic solvents. So it's not a great technology for organic solvents. So I do recognize those solvents, acetonitrile and DMSO, dimethyl sulfoxide. They're pretty aggressive. I've got a suspicion that you'll destroy those electrodes in some seconds, let's say. So I'm sorry to be disappointing on that. I just want to say thank you. If you do have any questions, do reach out to us. I have to take somebody to soccer practice now, so my apologies, but thanks so much for coming. Give me the most valuable thing you have, which is time, and it's appreciated. Okay, take care, guys. Have a good one. Bye-bye.